Coronavirus disease 2019 or COVID-19 caused by the virus SARS-CoV-2. Initial outbreak was first identified in December 2019 in Wuhan, China, and subsequently causing worldwide pandemic. As of 17 August 2020, there are more than 21 million confirmed cases, including more than 76,000 death cases being reported. COVID-19 can be transmitted through small droplets produced during coughing or sneezing, close contact with infected individuals, as well as fomic transmission. Age, gender, ethnicity, physiological condition, as well as genetic makeup of an individual can affect their susceptibility to COVID-19 infection. Currently, there are several supportive treatments provided to the patients such as mechanical ventilation, remdesivir, and dexamethasone. However, there is no any treatment that can be used specifically to eliminate SARS-CoV-2. Thus, GWAS study is essential to help in identify how genetics affect the susceptibility, resistance, and disease outcome of COVID-19, as well as help in the development of treatment for this pandemic. First, ethical approval was obtained prior to commencement of the study. Subjects will be collected based on the inclusion and exclusion criteria, where control group will be HIV-free and tested negative to COVID-19 individual, while case group will be patient diagnosed positive to COVID-19. Sample size was calculated based on this formula. Venipuncture was performed to collect the blood samples into EDTA tube, and the samples will proceed with DNA extraction using QIAGEN DNA extraction kit. The extracted DNA samples were measured using Nanodrop 2000 spectrophotometer and agarose jaw electrophoresis for their concentration and purity. Only those samples with purity between 1.8 to 2 and without presence of unspecific band after jaw electrophoresis will proceed with genotyping. Affymetrix genome-wide human SMP array 6.0 was the selected platform for genotyping, as it includes more than 94,000 probes to detect copy number variants, as well as more than 90,000 SNPs with call rate more than 97%. DNA samples were digested with restriction enzyme NSP1 and STY1. After digestion, the DNA fragment is ligated to an adapter which later serves as primer for amplification. The labeled DNA strand was then hybridized to the array for genotyping. The software Affymetrix Genotyping Console 2.0 was used to determine the genotype call for each sample. Data analysis and quality control was performed using Plink to check and clean the raw data. Samples with high missing call rate, low MNF value, related individual, and deviate from Hardy-Weinberg equilibrium was eliminated. Impute 2 was used to perform genotype imputation to predict the genotype at the SNP which is not directly genotyped. Manhattan plot was plotted to show the association of particular SNP with COVID-19. Throughout the study, polymorphism in ACE2 was expected to be associated with the susceptibility towards SARS-CoV-2, as ACE2 receptors serve as the receptor for entry of the virus. Polymorphism in ACE2 may result in altered ACE2 receptor which do not permit the entry of SARS-CoV-2. Other than that, TMPRSS2 polymorphism were also expected to be associated with susceptibility of COVID-19. Occurrence of polymorphisms may result in defective TMPRSS2 and unable to undergo proteolytic activity, which then entry of virus particle into whole cell is blocked. The locus 3p21.31, which comprised of 6 genes, were expected to be associated with the susceptibility, resistance, and disease outcome of COVID-19. SNPs in this locus were found to be associated with reduced expression of CXCR2 and increased expression of SLC6A2, and the frequency of risk allele for this SNP were found to be higher in patients receiving mechanical ventilation compared to patients with oxygen supplementation only. 3 SNPs found in locus 3q24.2, which affecting the blood group of an individual, were also expected to be associated with susceptibility to COVID-19. It was expected that individuals with blood group A is more susceptible to COVID-19 compared to individuals with blood group O. In a nutshell, GWAS study helped us to understand the susceptibility, resistance, and disease complication between genetic variation and COVID-19.